Introducing Gareth Hawkes from the University of Exeter Medical School, who's going to be talking about a recently published paper in Diabetologia. Gareth, in your study, you're looking at how people's BMI at different time points influences their later risk for diabetes and blood glucose control. Could you tell me a little bit of your background to that study and why you're interested in looking at this question?
Yeah, thanks for having me. So the background is that I've come from a maths background and I translated into the medical school world under some funding called Sophia. So we're looking at stratification of obesity by phenotypes. So we're really trying to pin down obesity and glucose diabetes phenotypes and stratify people by their specific phenotype rather than grouping everyone together. And what we were really interested in this particular question is because so there was a paper in 2020 that came out by Tom Richardson et al., where they looked at a similar question. But when we read it, we thought we can see a way of improving the statistical methods, just in terms of how well measured childhood BMI was in their study. So we thought, oh, okay, we can take it to the next level. And they found some interesting stuff, but we thought there was a glimmer of more there, so we tried to push forward.
Yeah, oh, that's very interesting, interesting start. And then you've used some quite large cohorts, haven't you, for this one, the UK Biobank, which is something I know my data's in there somewhere, along with thousands of other people, and then also a 1958 National Child Development Study. Could you tell me a little bit about those two cohorts and how they're used to answer medical questions of this kind?
Yeah, sure. So the UK Biobank is obviously one of the biggest of its kind. So when you go to even going to an American conference about genetics, everyone's talking about the UK Biobank.
Yeah, that's
so
great. Something we do well.
Yeah, yeah. So we have 500,000 individuals in the UK Biobank who have genetic data. So that's most common genetic variants have been measured, as well as an enormous amount of data about their lifestyles, their health, measures of, like you say, glucose, all the hormones, etc. It does have some biases in terms of it tends to be enriched for healthier individuals. But because of its size, you just can't not use it almost. So the reason we use the 1958 cohort, which is a lot smaller, so that's only 5,000 individuals with full genetic data. The reason we use that is to anchor the UK Biobank data in childhood. So obviously the UK Biobank doesn't have measures of those people when they were children. They're about age 45 when they joined. But the 1958 birth cohort, they were growing up at roughly the same time as the UK Biobank participants. So we thought it would be a really good way to anchor the UK Biobank data to a realistic societal representation.
Yeah, that's interesting. Yes. So, and then you've used a tool which we're seeing increasingly in diabetes research, which I don't know if this is a slightly harder question, but whether you can explain Mendelian randomization, which is we're seeing increasingly papers on Mendelian randomization so that somebody who's a non-geneticist like me or a non-mathematician can understand it.
Yeah, so the basic idea behind Mendelian randomization is we want to determine whether an exposure, so that can be anything, so an event effectively, is causal, so it actually causes the later on outcome, in this case, BMI and diabetes-related traits. The reason we use it is because it bypasses a lot of possible confusion that can happen in real data. So if you imagine you measured someone's BMI and you measured their glucose, the actual raw values, and you tried to say, okay, is A correlated with B? BMI can be confounded by a lot of external factors, such as smoking status, that you may not want to be being included in your analysis.
Or demographics, perhaps.
Exactly, absolutely, yeah. Loads of stuff. So what Mendelian randomization does is use the genetics, because your genetics are fixed at birth. What you're saying is these genetics can't have been influenced by later on other exposures that we're not interested in. And similarly for the outcome, so in this case diabetes, it can't have been influenced by things that happen during your life we're not interested in. So it's just a way of making a really clear causal pathway.
So you use Mendelian randomization and looking at childhood BMI and how that influences later diabetes or later glucose control. So what were your main findings?
I'd say the main finding is that it appears as though higher BMI in childhood, so larger children, appear to have a more a healthier insulin um, state of life in adulthood. So they've got higher insulin sensitivity and lower secretion. So their body is able to do more with less insulin than other adults of a similar age. The biological reason behind that we're not 100% sure about yet. So we have some hypotheses in that it might be due to when you're a child, you're able to make more adipocyte cells versus when you're in adulthood, if you put on more weight and you're as an adult, you're sort of fixed with what you had as a child.
Yeah, your adipocytes grow, don't they? But they
Exactly.
don't tend to increase in number. Mm.
Yes, exactly. And we're thinking that perhaps if it happens in childhood, you can have more than if you would have if you'd been a skinny child or a thinner child. And it's the stress that the adipocytes that go through that can influence your insulin state.
So is that quite a surprising finding? Were you expecting to see something different?
We weren't expecting it to be so consistent across all of the insulin measures. So in the Richardson paper that I talked about, they found a tiny hint of a protective effect of childhood BMI on type 2 diabetes in adulthood. So we thought we've improved the statistical method a bit. We'll take some more precise measures. So when you're doing a binary trait like diabetes, your statistical power is reduced. So we decided to look at more continuous traits like insulin sensitivity index and fasting glucose, etc. We weren't expecting it to be so consistently protective. And it's as we say in the paper, we're not sure what we should do with this information yet.
Yeah, you do say in the paper that you don't think it's ready yet to influence public health policy. So presumably you've got a few ideas of things that you want to do yourselves, or you think that other groups might need to do to so that we can understand a bit better what's going on.
Exactly, yes. So it wouldn't be right to say that larger children end up being healthier adults. It's just this very specific part of adulthood that's potentially protective, because you're not thinking about all the other diseases that could be much higher risk because of being a larger child, like heart disease, for instance. In terms of narrowing down what's actually happening, we're definitely starting to look into whether we can find some functional data, so actual measures of adipocyte size in childhood and adulthood for children who were larger in childhood, as well as just try to narrow down the specific genes that may be causing this time differential effect.
Yeah. That sounds very interesting. Well, I'm wishing you the very best of luck with your future research, and also looking forward to seeing your diabetology paper online. And thank you very much for talking to me.
Thanks for having me. Nice to see you.
Thank you.
Bye.